Welcome to another edition of Inside Humane Medicine Podcast. I'm Ollie Ford, the Dean and Chief Academic Officer of the University of Miami Leonard M. Miller School of Medicine. Today, we're diving into the groundbreaking research that could transform how we treat devastating autoimmune diseases such as systemic lupus erythematosus, which is also known as SLE or lupus for short, type 1 diabetes mellitus, as well as others. And to help us navigate through the complex world of immune regulation, we have invited a pioneer in the field of immunology, Dr. Thomas Malek, Chair of the Department of Microbiology and Immunology at the University of Miami Miller School of Medicine. Dr. Malek, Tom, welcome.
Yeah, thanks for having me.
And thank you for agreeing to elucidate the complex world of immune regulation and also delve into autoimmune diseases. But before we start, please share a little bit about your background with the audience, how your interest in immunology evolved, and that ultimately led you to become this preeminent scientist and chair of the department.
My background started in 1972 when I completed my bachelor's degree at Marquette University. And I couldn't get into medical school. And I go, well, I have a degree in biology. So I looked for a job, and I ended up getting a job as a lab technician at the University of Illinois Medical Center in Chicago. And it turned out to be an immunology lab. And I was actually fortunate when I was there to then stay in that lab and go on and get my PhD. And
at that time, immunology was just a pretty new field, wasn't it?
It was in the beginning. My initial project as a graduate student was to study the structure of antibody molecules, because a big question in the field at that time was, how could you generate so many antibodies against so many different viruses and bacteria with the limited amount of genetic material to code for them? And the way we were doing it was never really going to solve the problem. And when I finished as a student, actually, that problem was solved by determining that the gene organization for immunoglobulins was very complex, and that actually it could basically move around or jump around to create huge diversity. And, that's an important aspect of the immune system. It's, the immune system's like the nervous system. It has to really perceive things that it doesn't know it's going to encounter. And it does that, we understand now at a very detailed molecular way by specific antibodies and specific receptors that can perceive that.
So let's go back. Let's continue in that. You're this young scientist, and you are turning adversity into opportunity here. Didn't get into med school, but you get into this brand new field, and you're trying to chart your course forward, your path forward. What were the pivotal moments that led you to the line of research that you decided?
Well, I think so. This actually had to do with my choice for postdoctoral training. So my mentor was saying, well, you need to go to these labs that are studying gene structure. And I go,
and why was that?
Because of recombinant DNA technology. It was obviously, she was right that in terms of that was going to be a huge field going forward. But I was kind of interested to understand how the cells of the immune system worked.
How they talk
to each other and how they cross-regulate each other. And this was, basically virgin territory. So I chose a lab at WashU. And then later I went to the NIH, where I spent 6 years, where basically over that time I learned a lot about T cells and how T cells worked. And then when I was a postdoc at the NIH, I just was involved in discovering one of the subunits for the interleukin-2 receptor, which turns out to be a crucial sort of hormone-like, they call it a cytokine, but it's a hormone-like molecule. It's, there's many of these in the immune system. This still is one of the most important cytokines in terms of regulating the T cell compartment.
Very well said. We're going to put that in the parking lot. We're going to come back to that later. Yeah. Because I think right now it's important, before we start talking about your seminal work addressing the vexing problem, autoimmune diseases such as type 1 diabetes and lupus, it's important for the audience to get a little primer on the immune system. How does it work? And what are the cells involved? And in particular, the importance of T cells, because then we'll tie it all with the IL-2 that's in the parking lot for now.
Mm-hmm. So in terms of the immune system, there's three really crucial cell populations. There's B cells, which produce antibodies. There's antigen-presenting cells that present foreign
The bad actors. The H
from
foreign
the
agent.
virus and from the bacteria. And then there's the T cells, and the T cells really depend on these antigen-presenting cells to see antigen.
Okay. So the antigen-presenting cells presents the antigen to the T cell. They talk together via a receptor. And then what happens?
Well, the T cell interacts with the B cell, and then the B cell makes the specific antibodies. And the B cells independently can see antigen. So the B cell sees antigen, the T cells see antigen on these antigen-presenting cells, and then they work together to make your antibody response. And
what's that response?
So a key thing is there's very few cells in your immune system specific against any one particular virus, for example. So those cells have to replicate and expand in sufficient numbers to get rid of the invader. And, this usually takes 7 to 10 days. That's why when you get an infection, you feel sick for 7 to 10 days until your immune system is able to get rid of the virus. Especially on your first exposure, like COVID-19, when none of us were exposed to COVID. When you got sick, you were sick for several weeks, and for many people, the immune system then took care of the infection.
So antigen-presenting cells presents the foreign agent, the bad actor, to the T cells, B cells, then they start proliferating, making more cells to fight all the agents, all the bad agents.
Correct. But then there's
but they're also producing those things we call cytokines.
Right. Well, the cytokines are crucial for delivering signals to help regulate the B cell response and the T cell response, because once the response gets started, it also needs to be limited. Because if you have an unchecked immune system, you end up with a big inflammatory response that could actually be deadly. And that's actually also, if you go back to COVID-19, some individuals had such strong immune responses that was the reason for a lot of the pathology.
So very fascinating. So the body mounts this exuberant, this very large response to fight off the antigen, but at the same time, if that continues unchecked, then it can overwhelm the body.
Right.
So there's got to be a way to turn it off and to achieve that balance. Is that right?
Well, there's a lot of mechanisms to do that. I mean, we could talk about one specifically, which is immune checkpoints, because immune checkpoints are molecules, especially on T cells, that when they're engaged, they turn off the T cell response.
That's the turn-off valve for the inflammatory response.
And the reason I bring this up is in cancer, people have devised ways to block this checkpoint, so they'll allow your immune response to be reinitiated. And it's used in cancer immunology.
But that's because cancer cells are smart enough to turn it off.
Right.
So they can proliferate. But then if you can restore that immune checkpoint, then your cells can attack the cancer. So that checkpoint concept is important to control inflammation, but it's also important in a way to make sure cancer no longer evades the regulatory aspects of our immune system.
Yeah, I mean, it's sort of a natural mechanism to turn off T cells. So if you block that mechanism, you can help extend them, and that's really the basis of cancer immunotherapy, which many people are used today.
So now that the audience fully understands the immune system, now we can begin to address this concept of autoimmune diseases. You've talked about the immune system, its importance in fighting infection, or even cancer for that matter, but also recognize the fact that if it goes unchecked, then it can overwhelm the body, as in the case of patients with COVID-19 initially, and then who died because of overwhelming, inflammatory response. And so now let's talk about autoimmune diseases. What exactly do we mean by that?
So I think then we need one more primer about the immune system. So when you're developing your immune system, you're developing cells with many receptors, of which some can see virus. But also this all happens at random, so some of these receptors can also see self or your own tissue. And the immune system goes through elaborate mechanisms to shut down those cells. In the case of T cells, which I work on the most, an important mechanism is when they develop, if they see your own tissue antigens, they end up dying. And so the immune system purges many of these cells.
Right. So that they don't attack you.
Yeah. So I mean, they're gone. They could basically they're eliminated. But the process is.
So let's stop for a second. So for the layperson, basically those cells, when they recognize that, everybody else belongs, so there's no role for me, so I can just check myself out. I can just die.
Right.
Mhm.
But then this process isn't perfect. So actually some of these self-reactive cells get out into the circulation, and everybody has these. So and then these are kept in check by other immune mechanisms, but probably the most important one is a mechanism called regulatory T cells.
So those are the police.
Yeah.
Okay, they are policing to make sure that cells don't go after the body.
Right. So if you have a self-reactive T cell, these Tregs is what we call them. They're actively suppressing them. So that's under normal. So if you're healthy, everything is fine. But this process sometimes breaks down, and when it breaks down, you end up having some of your cells attacking your own tissues, and that's what really is the beginning of an autoimmune disease or an autoimmune attack.
The regulatory T cells are like police officers and make sure that you don't commit traffic violation or something.
Yeah, that would be one way.
Right?
Yeah.
Okay. So then to bring together the whole notion of autoimmune diseases, you developed that when somehow the regulatory T cells were not effective in preventing those T cells that are self-reactive from attacking the body. Is that what I'm understanding?
Well, I mean, I think basically that would be correct. I mean, there can be other reasons besides the Tregs, but basically when you have autoimmune disease, immune mechanisms are failing, controlling, silencing.
Right.
The police went to the diner to have donuts that day or whatever.
They were not looking.
They weren't looking. And then, and I think the thing about autoimmunity, it's like cancer. Over almost any tissue in your body can be attacked by the immune system. And there's certain tissues certain autoimmune diseases that are more common,
So is there some trigger that causes those T cells, which you call self-reactive T cells, to actually attack the body?
Right. So I think there's two theories. Well, I don't even think it's a theory. One is we know that there's genetic variants or genetic predisposition for some individuals where they're more likely to get an autoimmune attack. But there also seems to be some sort of environmental trigger, and it's often thought to be an infection, but it's difficult. But usually they're not linked in time. So you can have an infection today, and it can do things to your immune system, and then the autoimmunity shows up 6 months later, a year later, several years later. So it's been difficult to kind of prove that those kind of associations. But when they look at identical twins that have the same genetic makeup, one twin will come down with autoimmunity, the other won't. So I think that's certainly evidence that there has to be some interaction with the environment that's important to come down with an autoimmune attack.
An excellent point. So how prevalent are these autoimmune diseases globally?
Well, in the United States, they estimate 20 million people have a various autoimmune disease. So that's about 5% of the population. And I think, you could probably extrapolate that to, worldwide.
Yeah, and why is it important to treat autoimmune disease?
Well, I mean, eventually it degrades your tissues. It can cause a lot of problems with day-to-day living. And for some individuals, it ends up being, lethal.
And on a personal note, my sister's life was devastated by lupus. So I'm very, very interested in this particular problem, and it may have actually pushed me towards my own interest in immunology. So thank you. And now let's talk about this situation. So in a healthy immune system, and we already mentioned it. It's like we're running a tightrope. On the one hand, we want to make sure we activate the immune system to fight microbes and foreign agents, but at the same time, we do not want to go overboard or let it go unchecked, because, as you said, it could have adverse impact on the body and lead to destruction of the normal tissues. So tell us how tightly regulated this dance is and the critical parts that control that dance. I know you already talked about the Treg. What else should we be considering?
Well, I think part of the immune system is actually programmed. Once the cells start expanding, there's genetic programs in place to sort of limit their overall expansion. Of course, if they're continually being stimulated, that can, if they're not effectively getting rid of the virus or bacteria, that process won't necessarily be sufficient. And then there's a whole host, actually it's a very complex set of mediators. One that we talked about is these immune checkpoints for T cells. There's also cytokines, other hormones the immune system makes sort of as a check to sort of turn down inflammation. So as the immune response expands, they can make mediators that help tune down the immune system. There's it's mentioned interleukin-2, but I think we're up to interleukin-35. So there's many mediators involved.
But coming back to, we alluded to the role of the regulatory T cells as almost police officers. And now, is there a way to selectively target that population so you can produce more police officers to monitor this process and prevent it from going awry?
So there's two approaches that are being taken in the field. And one of these goes back to interleukin-2. And then the other really goes to where people are trying to grow these cells in culture and reinfuse them back into patients. And that's both of these are under intense clinical development by big pharma and biotech companies.
Let's talk about your contribution.
Okay.
What your approach?
Yes.
What you found and why you decided to go there.
So let's just give a short primer about interleukin-2. So interleukin-2 was...
Now we can take it out of the parking lot.
Yeah.
Okay.
So interleukin-2 was discovered in the early 1980s as a molecule made by T cells that get activated during an infection. And when it was discovered as T cell growth factor. So it has an important properties actually in causing the expansion of your cells that you need to fight off a virus. And this was studied intensively for a long period of time. And in fact, the first drug for cancer immunotherapy was interleukin-2, based on the concept that it was important in expanding these your sort of T effector cells, which was used to kind of get your anti-tumor T cells expanded. Now,
the activated T cells.
So one experiment that was done in the early 90s that was kind of perplexing is in mice, they says, well, let's get rid of the IL-2 gene, right? So the prediction would be, we get rid of the IL-2 gene, the mice are going to be immunodeficient, the immune system isn't going to work anymore. And what was found was the complete opposite result, is that the mice had uncontrolled proliferation of their T cells, and then they were dying of a systemic autoimmune disease. So this was this was perplexing in the field, and there was a lot of theories proposed on why this might be the case. And this is sort of where, our work started to turn toward Tregs. Is, when I came to the University of Miami in the 80s, I worked on IL-2 as a postdoc. And then I came here, I was working on IL-2 and some other projects, more looking at T cell activation. And I wanted to, we were doing this, studying, in vitro experiments and molecular experiments, and I wanted to know how IL-2 works physiologically. So we went on to make mice that were genetically engineered, but just to study IL-2 signaling in a physiologic context. And what we learned was that what IL-2 also does, it's actually essential for these regulatory T cells. So when you knock out IL-2 signaling, the first thing happens is these Tregs don't develop. And when the Tregs don't develop, these self-reactive cells that the police are normally doing a good job, now they're basically none of them are there. So the immune system has carte blanche on attacking your tissue.
So there's looting throughout the city.
Right.
Okay, so there's destruction.
Right.
And hence the importance of IL-2 in also developing the population of regulatory T cells.
So IL-2 has two crucial things. One, making and maintaining your Treg population, and then also boosting the effector compartment. So
how are you going to exploit this paradoxical role of IL-2 in inflammation in order for you to actually combat autoimmune disease?
So basically, low IL-2 signaling is very productive for Tregs, but not for your immune cells that need to expand.
Correct.
So the field actually has started exploiting that concept by using low dose interleukin-2. So, with gene cloning, all these molecules we work with have been cloned, and then they've been used in the clinic for the last about 15 years, and they can cause Tregs to increase sort of selectively in most individuals that get it, and there's been some clinical improvement. But the issue with recombinant IL-2 is that it has poor pharmacokinetics, poor pharmacodynamics, so it's really not a good drug for this purpose. And
So what do you mean by that? So you just basically it doesn't last so long.
Yeah, basically, if you inject it into a person, it lasts minutes to hours. So to get this to work well, you need to be basically continually infusing IL-2.
So let's talk about it. So we know, therefore, that if we can selectively regulate the Treg population, then that could help fight or keep those suppressed or those self-reactive T cells under check. But the challenge is the low dose IL-2 that you'd have to give, it doesn't last long enough. It doesn't stay around long enough. So that's the challenge.
Yeah, right.
And but you came up with a way to overcome this challenge because you understand the importance of giving low dose IL-2 to help expand or continue to maintain the T regulatory population. Tell us about that.
I decided that I want to take some approaches to try to develop a better form of IL-2 that was longer lasting and would be more effective. And we sort of contemplated several strategies. One strategy that was used by others is to bind IL-2 to antibodies to IL-2, and that complex could stimulate Tregs or T effector cells depending upon how it was displayed. So the area we pursued was to link IL-2 to one of the receptor subunits for IL-2, kind of as a surrogate antibody, where it could interact and then stimulate the Treg compartment. And we learned that actually when we did this, there were several predictions, and it turned out that IL-2 interacts with the receptor subunit and it stimulates the Tregs very well, but it does it in a very complex way.
But it delivers it in a slower way, and it's more sustained.
Right. So
which is important, right?
Yeah. So the way it interacts with the IL-2 receptor subunit is sort of as the carrier, is it interacts in a way basically to inactivate it. It binds in a way because what we call transdimers. So the transdimers are inactive, but then some of it slowly releases into a monomeric form. So basically you could consider two IL-2 molecules. We call this IL-2 CD25. Two of them are binding to each other. That's the inactive form, but then it can be released based on sort of the biochemistry of the molecules. And this is by controlling the concentration of this IL-2 CD25, you can selectively keep the concentration low enough to target the Tregs. And the real advantage is that you can cause a continual slow release of this IL-2 activity for a long period of time. So it's not just increasing the half-life, but it's actually increasing what we've learned recently.
Steady state.
Yeah, it's also more efficiently inducing IL-2 signals in the Tregs.
And the consequence of that for autoimmune diseases is?
The consequence is that in the model systems we've been working with, we can achieve complete control of autoimmunity, and we've been using a model of type 1 diabetes. And we like that model because the model develops spontaneously with genetic risk factors like human disease.
And it stops it. It stops the type.
As long as we keep them on the drug.
That's an important breakthrough. How did you feel when you recognized that this new biologic construct IL-2 CD25 achieved what you thought it was important?
First thing I knew, like for the first experiment we did, I knew it worked really well targeting the Tregs. So that in itself was really really exciting. And then and then I was able to attract interest by big pharma to develop it. So
how'd you do that?
Well, so this is a little interesting story. I was through tech transfer era, we were trying to license it to different companies. And I was at a meeting, a T-Reg meeting in China, and some guy walked by me and he had on his badge, it said Bristol Myers. So I said, I will talk to him. Maybe he's interested. And so I talked to him and he went back. And then a month or so later, I got invited by Bristol Myers to present our results. And then after they, tested the molecule themselves to ensure that it worked, we entered a licensing agreement with them, which is maybe about 8-9 years ago.
And now you're in clinical trial.
So now it's in
phase 2?
Phase 1B in lupus, SLE, which we talked about earlier, and atopic dermatitis.
Fantastic. So your work could potentially bring a cure to this vexing problem that personally affected my family. So I'm quite jazzed about it and grateful to you.
Yeah, well, we're just hoping it continues to be able to move through the system.
So what are some other evolving therapies in the field of autoimmunity that get you excited at this point?
Well, I mean, I guess the thing to maybe go back to is if you have autoimmune disease now, how do they treat it? And they usually give it drugs that broadly turn off the immune system for the most part,
treat, which could be bad for
treat the symptoms for the most case. And if you're on those drugs for a long time, your immune system suppressed, you have problems with infections. Also, if you're on them for a long time, they have certain toxicity profiles. So, what's still exciting to me, this is why I'm still working, because, my wife goes, when are you going to retire? So this is true. And that the reason I'm still working is the concept of Tregs is really been boosting them, with IL-2 or by adoptive cellular therapy. The goal is to kind of reinstate the normal physiologic state that kept those cells in check.
So it's a recalibration. Is that
recalibrate the immune system or reset it back to a time before you were having
trouble?
Right.
That's amazing. That is amazing. Before it went awry, now you want to bring it back to its original state.
Yeah.
Press the reset button.
Yeah.
Fantastic.
And I think that's why there's a lot of, there's a lot of interest in the academic and the industrial community, because it's really a new concept. Well,
if you could do that, that would be remarkable.
Yeah.
And I can see that's why that keeps you going. So in closing, I know we're a little bit over time. What message do you have to young scientists coming up as far as their dreams and their approach to being impactful?
I mean, well, I like to tell students that are interested, new students come in, there's so much noise. They're worried about their exams, their classes, when they're going to graduate, what jobs they're going to get, when they're going to get grants. I tell them if they can focus on the science and move the science forward, all these other things are just fall into place, so that's a message I like to give to a lot of the graduate students that enter the program. And
Dr. Malek, you're an amazing role model, an inspiration to all, and certainly one who's been quite impactful. And it is my sincere hope that this clinical trial that's taking place with IL-2 CD25 biologic will prove to be the panacea that we've been looking for to control all these autoimmune diseases. And it will have been all because of you. So thank you for your career. Thank you for all the things that you've done to improve human suffering. And thank you for being our guest today in the studio.
Oh, thanks a lot.
It's been
fun to do it.
Okay, thank you. And this has been another episode of Inside Your Miami Medicine. Looking forward to seeing you again for the next podcast.